everyone. Today we come to you with a special news story on autism spectrum disorder, or ASD. Sit back and get ready to listen because we are about to share everything you need to know about ASD and share some groundbreaking research results in the field. We will then conclude this special feature with experts who will explain an approach they are developing in order to further investigate the disorder. Let's first start by understanding the disease. Today we have Dr. Jennings with us who will provide us with some background on the disorder. Starting with the causes, what can you tell us, Doc?
Yes, thank you for having me. So basically, autism spectrum disorder has no single known cause, and due to the complexity of the disorder, and since the degree of the symptoms varies, there are likely numerous causes of ASD. Both genetic and environmental factors may play a role. Several different genes seem to be associated with ASD, and there are mutations that can occur in genes of some ASD cases. Some of these mutations can be inherited or spontaneous. Genes implicated in ASD may have an effect on brain development or brain cell communication, or they may determine the severity of the symptoms. So far, researchers are currently studying whether viral infections, medications, or complications during pregnancy, or even air pollutants can trigger autism spectrum disorder. Also important to mention is that scientifically speaking, there are no proven links between ASD and vaccines. Certain factors may increase a child's risk of developing ASD, and these include a child's sex, seeing that boys are 4 times more likely to develop the disorder, family histories, and other disorders such as Fragile X syndrome, tuberous sclerosis, and Rett syndrome.
Interesting. This really seems like a complex disorder. Can you tell us about the symptoms of ASD?
Definitely. ASD is classically associated with symptoms of impaired social communication, restricted interests, and repetitive behaviors. Affected individuals won't typically show all of the aforementioned symptoms, but most will show several of them. In terms of social communication, children and adults with autism may have difficulty with both verbal and nonverbal communication. For example, affected individuals may not understand spoken language, facial expression, the purpose of eye contact, and emotional cues. Also, they may not appropriately use gestures that express a tone of voice that matches that of the conversation. We also see restricted and repetitive behaviors that vary greatly across the spectrum. Such behaviors include repetitive body movements such as rocking, flapping, spinning, ritualistic behaviors such as lining up objects or repeatedly touching things, and also having a need for unvarying routine. In other words, they're highly resistant to change and have an intrinsic need to follow a specific schedule, eat certain meals, follow certain routes, and etc. These symptoms of autism may begin early in childhood, persist, and continue to interfere with an individual's daily activities.
Wow, so clearly there are a number of symptoms for this disorder. What implications does that have in terms of diagnosing ASD? How is that done usually?
So far, the Centers for Disease Control and Prevention, or the CDC, have identified behavioral patterns for autism spectrum disorder in young children. And this includes not responding to their name by 12 months of age, not pointing to objects to show interest by 14 months, not playing pretend games by 18 months, avoiding eye contact or preferring to be alone. There's getting upset by minor changes, flapping their hands in those repetitive movements, and having unusual and sometimes intense reactions to the way things smell, feel, taste, or look. It's important that parents must first notice these changes in their child's behavior, then a diagnostic evaluation would be performed. And this typically involves an interview and play based testing with a child who's by a psychologist, developmental behavioral pediatrician, child psychiatrist, or other healthcare providers.
So I'm getting that in order to diagnose the disease, the parents must first notice certain behaviors in their children. What happens from there if there is an ASD diagnosis? Are there any treatment methods currently being used to target the disorder?
So, there are currently no cures to ASD. However, there are many interventions to help reduce the symptoms of the disorder and improve cognitive ability and daily functioning. There are behavioral interventional strategies that focus on social communication skills, minimizing limited interests and repetitive behaviors characteristic of ASD. We can actually split these interventions into the following categories. There's behavioral and communication approaches, which involve social skills training, speech therapy, occupational therapy, and enforcing positive behavior while discouraging negative behaviors. There are also dietary approaches where changing one's diet may help the symptoms, and there's also medications for certain symptoms of ASD can also help, such as anti-anxiety medications, selective serotonin reuptake inhibitors, antipsychotics, anticonvulsants, and stimulants to address the potential symptoms of ASD. These interventions can drastically improve the daily life for those with ASD by minimizing their symptoms, of course.
Thank you, Dr. Jennings. Next, we have our research highlight with Dr. State. Thank you for taking time out of your busy day to give us a bit of an overview about the recent paper about ASD that you published.
Yes, of course, no problem. In this recent 2020 paper, we've really found some interesting results. Very briefly, we started off our experiments by looking for 10 major ASD-linked genes to study, mutating them and observing their effects on the brain of a Xenopus tropicalis model. We then searched for a potential drug compound that could rescue some of the negative effects we observed. The final steps in this study involved us trying to apply these Xenopus results to humans by performing human neural progenitor cells and network connectivity studies.
You mentioned that you used the Xenopus model instead of the mouse model we are accustomed to hearing about. Why did your team choose this?
Yes, that is correct. We chose to use a model of Xenopus, which is a type of frog. This model allowed us to perform something called unilateral mutagenesis by CRISPR-Cas9 injection, in which we commutate frog genes on only half of the brain and then compare the effects to the unmutated side. Therefore, a single frog gave us experimental and control results, which allowed us to detect very subtle changes in brain size. Xenopus has other key advantages for this purpose, such as relatively high throughput and low cost for an in vivo model.
That is very interesting. Moving on, you mentioned that you studied some candidate genes that may be involved with ASD. We would like to know why you chose to study the 10 genes and what you discovered.
The 10 genes that we chose to study are not only ASD-associated genes, but they are also expressed during neurogenesis in the Xenopus model. Disruption of these genes will actually lead to changes in cell proliferation by increasing the ratio of neural progenitor cells, or NPC, leading to changes in the size of the brain, more specifically, the teleencephalon. This was not observed in our control brain with genes not associated with ASD, which means that there's a link of neurogenesis causing an increase of size in the brain with ASD-related genes.
So now would you be able to elaborate further on how genetics has an effect on brain growth and development?
Yes, so there are some genes that are considered ASD risk genes, and we discovered that they converge with cortical neurons within the inner subventricular zone while the brain develops. These points of convergence will help us and future researchers identify and explain mechanisms and the cells involved that are associated with ASD. Also, this inner subventricular zone contains highly proliferative cells, meaning that alterations may change the size of the brain.
At some point in the study, you performed a drug screen. Can you briefly explain to us what you were hoping to find through the screen and if you were successful?
We performed an oncology drug screen in the hopes of understanding the molecular mechanisms underlying this NPC phenotype and to identify any general resilience factors to ASD risk genes with the potential to lead to future therapeutic treatments. So during the drug screen, we discovered that estrogen actually acts as a suppressor of the ASD phenotype. So estrogen protects against mutations in ASD risk genes by modulating neurogenesis rather than rescuing gene deficits. We then found that estrogen signaling plays a crucial role in neurogenesis by opposing sonic hedgehog signaling, which plays an important role in regulating neural proliferation and maturation during brain development.
So now a question we all want to know is whether these estrogen results you found are applicable to humans.
Yes, that's exactly what we tried to find out. So knowing that estrogen signaling is required for teleencephalic neurogenesis and inhibits SHH signaling in humans, we looked to see if we could rescue ASD effects in human models. Using human 2D and 3D in vitro models of brain development, we found similar neurogenesis defects following mutations of the genes we studied, and estrogenic compounds were shown to successfully rescue this dysfunctional phenotype.
Now that we have discussed the paper, can you please help the viewers understand the implications of the work we have just talked about?
So first of all, the work I have shared with you shows how Xenopus could be used for a range of models supporting ASD systems, biological analyses, which could potentially be used in future studies. Moving on, we were also proud to have discovered a potential mechanism of resilience that may be impactful across a range of ASD risk genes. Our finding that estrogen suppresses a convergent ASD-related phenotype in both Xenopus and human cell culture brings up a really intriguing hypothesis regarding the origin of the well-established male predominance of the ASD phenotype. However, I believe the major implication of the study is that we identified a novel point of vulnerability in brain development. Specifically, this was the first time that we, as researchers, have shown the ASD effect on neurogenesis, as opposed to the previously confirmed effects on synapse and transcriptional regulation.
Thank you very much, Dr. State. We now have with us on call a group from the University of Ottawa's Translational and Molecular Medicine program, here with an innovative research proposal to further investigate ASD. So let's start off with talking about the inspiration behind this proposal.
For sure. So it's known so far that there is not one singular cause to the development of ASD. However, previous studies have shown that irregularities in the amounts of circulating sex hormones can contribute to the presentation of ASD and its symptoms. With estrogen specifically, it was shown by Dr. State that there is a suppression of the ASD phenotype. Estrogen is an important molecule playing important roles in the pathophysiology of neurodevelopmental disorders. So we decided to build on the study conducted by Dr. State and colleagues to further investigate the role of estrogen in ASD.
I cannot wait to learn more. May you please tell us about your research objectives? What do you hope to gain from this experiment?
Of course. So we will use a social mouse model, male fetuses in particular, to investigate the role of estrogen in contributing to autism spectrum disorder phenotypes, specifically complex social interactions and repetitive behaviors, and potentially develop therapeutics to treat its symptoms.
Unlike Dr. State's recent paper that used the Xenopus model, you plan to use a mouse model in the study of autism. Why did you make this decision?
That's a good question. A lot of current research surrounding ASD uses mouse models, which are manipulated with a causative agent to produce the neurodevelopmental disorder. We can establish a behavioral pattern of ASD in mice that closely resembles the behavior pattern we would expect to see in humans.
We want to specifically use the mus musculus, or the house mouse model, to study autism, as it is a social species that we can use to study impairments. Additionally, this particular mouse model exhibits compulsive grooming behavior, which is another distinctive symptom of ASD. And as such, a repetitive behavioral assay can be used to quantify this phenotype and again determine how estrogen contributes to it.
That sounds fascinating. What do you plan to test in terms of your experiment?
So we know that estrogen is highly expressed in several regions of the brain. We will be focusing on two of those. First, the cortex, which is responsible for higher order brain functions, including complex social interactions. And second, the cerebellum, which is responsible for the control of voluntary movements. We chose these two regions as we're hoping to investigate differences in sociability and repetitive behaviors.
To build off of that, we will first decrease the levels of estrogen in our mice by administering an estrogen production inhibitor, letrozole, in our experimental animals and a placebo in our controls. Then we will be using transgenic mice to specifically target estrogen receptors in our chosen brain regions. And by using LOX-P mice, we can knock out estrogen receptors to selectively study the impact in our specific brain regions. A LOX-P mouse contains two LOX-P sites that flank a genomic segment of interest known as the FLOX locus. In our experiment, this locus would be the ESR2 gene, which encodes the estrogen receptor ER-beta, and that's important in modulating neurobiological systems.
Now, you mentioned that you will be looking for differences in mouse sociability and repetitive behaviors after estrogen inhibition and estrogen receptor knockout. So how do you intend to measure and quantify such behaviors?
Yeah, so as mentioned earlier, the mouse model that we will be using engages in high levels of social behaviors. A variety of social assays have been described in the neuroscience literature that maximize the relevance of the assay to the types of social deficits that are specific to autism. We elect to use what's called a three-chambered social approach task, where sociability is defined as the mice spending more time in environments with other mice compared to environments with unfamiliar objects. All mice also exhibit spontaneous motor behavior, such as circling, jumping, and self-grooming, but these behaviors exist for unusually long periods of time in autism-induced mouse models, so we can use a timed assay to monitor these repetitive behaviors.
And generally speaking, what do you expect to see as outcomes to your experiments?
From what we know so far from past research and findings, we expect to see significant differences in experimental groups compared to control groups. Specifically, we expect to see that experimental animals would display smaller tendencies for social interactions and a larger portrayal of repetitive behaviors compared to the control animals. Of course, we always hope that our results will be in line with our objectives, but we can also expect that there will be some outliers, and so it will be essential to implement the use of statistical tests to prove the significance of our results.
And how do you see this research translating into a clinical setting? In other words, how do you feel your research will contribute to the understanding of autism and the development of potential therapeutics?
So we expect that estrogen plays a role to suppress autistic phenotypes, and as such, it may be the reason why males tend to have a greater risk of the disorder. We hope that our research will elucidate estrogen agonists as potential therapeutic options for the treatment and modulation of ASD.
Thank you very much, Ricky and Nadine. We wish you the best of luck and hope to see your results published in the future. That concludes our news highlight on autism spectrum disorder. Thank you for tuning in, and we will see you again soon.